Okay, so hey everyone, welcome to the MIT Biotech Group Entrepreneurship Initiative Fireside Chat series. I'm Elliot. I'm a fifth-year grad student in Rick Young's lab, and I'll be hosting today's chat. So thanks for coming. Thanks to Dr. Kevin Mills for joining us. I'll ask that everybody keeps their mic muted to avoid noise in the Zoom. And if you have questions for Kevin as the talk is going on, please put them in the chat. Or you can save them for the last 15 minutes or so, which we will reserve for audience questions. Depending on the number of questions, you might be able to unmute yourself and ask your question yourself. But otherwise, we're going to have Sam curating the questions from the chat and asking any questions that seem to be asked frequently or particularly relevant. Okay, so with that, it's my pleasure to welcome Dr. Kevin Mills back to MIT for this MBG event. Kevin earned his PhD at MIT with Lenny Guarente in 1999. He did a postdoc with Fred Alt at Harvard after that. And during his training, he established expertise in the fields of DNA repair and in cancer biology. So he then went on to start his own group at the Jackson Laboratory to study genome instability in cancer. So, and after several years of running his research group, Kevin discovered some key tumor biology that led to the founding of a company called Cyteir Therapeutics. And then after seeing Cyteir into clinical stage, Kevin moved on to a new role as the CSO of Civetta Therapeutics, which is a relatively new drug discovery startup. Kevin is an accomplished academic, having published more than 30 papers. He also has lived the experience of a biotech founder, having pitched to dozens of VCs and even having won a pitch contest here in Cambridge. I'm looking forward to hearing more about Kevin's journey. Kevin, I've purposely left out a lot of details so that you can fill it in for us. So why don't you start by just telling us about what it was like to get your PhD at MIT in the 90s?
Sure. Well, Elliot, thank you. And thanks to everybody for the invitation to participate in this fireside chat. It's a lot of fun to sort of be virtually back at MIT. It's been a while, and it's a lot of fun. So I really am thankful for the invite. What was it like when I got my PhD? So I was in Lenny Guarente's lab, 94 to 99, and at the time I was in his lab, he was his lab was just beginning to had just a few years before that begun to take the leap into the biology of aging. I mean, he really made the first part of his career in using the tools of genetics to study regulation of transcription, and then had a couple of really dynamic grad students that preceded me that came into the laboratory and wanted to go on this journey into using genetic tools to study aging, and I thought that sounded amazing. So when I came to MIT in '94, I was immediately drawn to the Guarente lab and to the science that they were doing and the questions they were trying to ask. It was a pretty cool time. It was, as an aside, I was also going to say, I was in the first or second class that was in building 68. So we had just moved into building 68. The biology department had just moved into building 68 when I joined, when I came to MIT. I think I even interviewed when it was still in building 57, I think. When I was there, if you sort of look out the windows, around the Kendall Square area, there's a lot of stuff that's there now that wasn't there when I was there. For example, that sort of quad right outside the building there used to be a parking lot where all the food trucks would park. That's where I got lunch every day. So a lot has changed.
There's still one food truck that comes there that's pretty good.
It used to be a lot of food trucks, and the line would be around the block. It was probably the best part of the day. But yeah, no, getting my PhD, at MIT, in '99 was terrific. Lenny's lab was amazing. The biology department was and still is this incredibly dynamic, creative, innovative place. And I sort of felt like there was no question that couldn't be asked, and for that matter, no question that couldn't be answered. And uh, and it was amazing because you could walk out the door, and in a 2-minute walk in any direction, there was a tool or an instrument or a resource or a collaborator that just made it like being like a kid in a candy shop to be able to do science, which was so cool as a grad student. Um, to be able to just sort of have the world of opportunities before you. And it's even more so now. Right? There's so much there. There's so much energy and so much activity. Um, COVID is a little bit too bad. That's changed the way we work, but that's a temporary thing. Um, so it was cool, right? The biology department was amazing. I was also there at a time before they were doing rotations, so we didn't actually even have an opportunity to sort of test drive any of the PIs before we joined the lab. So that was, it was an interesting time when the PIs were selling their labs as much as we were trying to get into the labs. But really, really, really pretty amazing.
Yeah, yeah. So you mentioned that a lot of the buildings that are there in Kendall Square now were not there back then. So I'm curious, you got to sort of see some of those buildings be built maybe during your time at MIT, or like at least see a lot of biotech come into existence. So I'm curious what the opinion of biotech, of of the grad students and of the biology department was at the time, right? Because I think MIT now has a reputation for being very open to biotech and really integrated into the scene relative to a lot of other programs. So I'm curious what that interface was like back then.
Yeah, that's a terrific question. I mean, um, so at the time that I was at MIT, the notion of a career in biotech was the farthest thing from my mind. Right? It was not something I thought about. It was not something that was kind of part of my future ambitions, and it wasn't as omnipresent as it is around the Kendall Square area now. I mean, certainly there was biotechnology there, and certainly the biology department, but maybe MIT more broadly, has a very strong entrepreneurial kind of attitude, entrepreneurial phenotype, more so than other academic institutions for sure. And so there was definitely activity around, and I was aware of it, right? I mean, Paul Schimmel, his laboratory was right next door to Lenny's at the time. And Paul has been kind of a serial entrepreneur, right? And at the time that I was in Lenny's lab, he was actually taking a step into that entrepreneurial direction as well and starting his first company, Elixir. And so it was definitely there. Not as much as it is now, right? Now you can sort of walk out the door and in any direction, you're going to bump into a biotech, a startup, a pharma company, or a VC in a 100-square-foot radius of building. And it wasn't quite like that, although it was beginning to be. And I think, we as grad students were aware of that and aware of that energy. But it hadn't matured to the degree that it has now. And as I say, for me, my ambitions were much more focused on academia than on industry. And I think that reflects a certain naivete that I had at the time about how far apart academic research and industry research are. I've since come to believe that the differences are minor, right? And the similarities are much stronger.
Interesting. I wonder if that is something that has happened recently. And back then, perhaps your perception was accurate, but that the biotech scene has changed, or that they've merged somewhat. Maybe academics have changed. Right.
I think there's no question that academics have become more industrial and more entrepreneurial over time, and that equally on the industry side of things, the kinds of research that are happening at least in small biotech labs is more academic in nature, perhaps, than it used to be.
Yeah.
There's a blurring of the line between those.
Yeah.
I think there used to probably be a mindset that industry, at least when I was in grad school, that industry equaled big pharma, right? And now there's clearly like another piece of the puzzle, which is small biotech, which is a very distinct kind of activity from big pharma or from academia in some ways. And so I think that, I think what's emerged over the years is that there's this other kind of thing called small biotech that's not the same as big pharma.
Yeah, for sure. And so you mentioned that you were sort of proximal to companies that were spinning out around you, like Elixir and others. So, I'm wondering, like, can you speak about, what sorts of things you saw the founders doing back then that, and maybe relate them to what you did in the future, and did any of those things that you saw sort of inform the way that you went about founding your own company?
Yeah, that's a terrific question. I mean, I wish the things that I had seen back then informed my own company foundation more than it did, perhaps. But I got to see a couple of companies get started kind of up close. Elixir was a company that Lenny Guarente started, and sort of around that time, maybe a couple of years later, David Sinclair founded Certrus, and David and I overlapped in Lenny's lab, and David's an old and dear friend of mine, and so I got to see the process play out a couple of times where, there was the venture capital piece of it, right? Trying to find the financial backing, trying to make the scientific case, trying to figure out the data package that would secure that financing, right? And try to really create a value proposition. I think it was different back then, maybe a little bit, than it is now in a couple of ways. One of those is that, there was a lot of activity. There was a lot of company foundation kind of around that time in the 90s, late 80s and the 90s. But there weren't a lot of great kind of tried and true business models. And so it was really based on the science and kind of the promise of where the science could go. And I saw that happen, right? And I saw how the data was being sort of portrayed to the venture community in a couple of ways. So fast forward that to my own company foundation, and I think, the life sciences venture community has learned a lot as well over the years. And there are some tried and true business models. There are some kind of paths to success for small spin out companies. There's some pattern matching that can be done now. And, had I looked back on those early experiences, I would have probably given me more of a roadmap of the things to do that are successful and the things to do maybe that are not so successful.
Yeah. And one thing that I would imagine is definitely different is the way that companies, there was probably no venture creation back then. Companies came from the scientists probably almost entirely, right? Because Flagship wasn't founded really until 2000, I think. And they really brought the venture creation model to fruition, right? Is that accurate with your impression?
Yeah, no, I think that's true, right? I mean, I think, the early days of life sciences venture, Venrock was a big player in this space, and has a long and successful history of starting up companies, but those have largely been, investigator founded companies, right? And not sort of the sort of venture driven kinds of things. But that model, has grown up and matured, and certainly in the Cambridge area for sure, with the Flagships and the Third Rocks of the world. And it's a really successful model, right? I mean, because they can incubate a company for a long period of time to mature the technology to a point where it makes sense to go out and sort of syndicate the deal or do it in a kind of solo venture kind of a deal.
Right.
But that kind of venture incubator kind of approach enables them to, kick the tires on a new technology for a long period of time to de-risk it in a way that can really set it up for success. And
Right.
I think part of that model, the other part of that model that's really valuable is part of the de-risking is not just doing the science, it's also getting the right team around the science in order to be able to prosecute it.
Right. So I want to move on to some of your research and the founding of Cyteir. So you open up your lab at Jackson Lab, and you start publishing papers and training people. So tell us about the research and the discoveries that happened during this time that led to the idea behind Cyteir.
Sure. Well, Elliot, you should remember some of those because, as you well know, you're on one of the key papers.
Right. That's
as a summer
pure
student.
coincidence. Pure coincidence for the record in the chat.
I think it's not pure coincidence. But yeah, so when I started the lab at Jax, I wasn't really thinking about the commercial implications of the research. But what I was doing was writing a lot of grants that were pitching the research as being relevant to human health and disease, right? There's always a line in there that this will help us discover new drugs, or this will help us develop new diagnostics, or this will help cancer patients in some way. And it always sat in the back of my mind that we never connected those dots, that we never really delivered on the promise that this work was going to help patients. And so there was an idea that I had while I was a postdoc that never really turned into a project in my postdoc lab, but was always something that was kind of in the back of my mind, and managed to kind of get it going a little bit in the laboratory at Jackson, and brought in some people around that, brought in a postdoc and a grad student around that, and a summer student around that idea to start to kind of test the idea a little bit. And lo and behold, the data actually looked really interesting, and turned out, without sort of torturing the data, that we discovered that we were able to very selectively kill certain classes of tumor cells using a genetic manipulation. And then we actually found a small molecule that could do the same thing. We actually looked for the small molecule not because of any drug discovery kind of ambitions, but really as to provide a research tool. We were looking for a chemical biology tool that would complement the genetic approaches we were taking. But once we discovered that small molecule could selectively and potently kill certain biomarker-defined classes of cancer, then the idea really was pretty obvious that this could turn into a therapeutic application. So then the question was, what do I do with that, right? Do we publish that and sort of let the world try to run with it? Do we try to put some intellectual property around that and license it to a pharma company? Or do we try to prove the principle through doing the work ourselves? And in an academic, especially a small nonprofit academic institution like Jax, it's really hard to do legit drug discovery, drug development program. And so as I sat down and thought about what are the different ways to deal with this information or sort of take this forward, one of the realizations that I came to was that if I wanted to do translational science, then I also had to do commercializable science, right? At least if you're in the therapeutic space, translating that science is in many ways equivalent to selling it, right? You've got to have a product if you want it to reach patients, if you want it to reach people. And so that was kind of when my thinking about the divide between academia and industry and biotech really changed. And so I took the decision that the best way to be a translational scientist and to be able to translate this work that we were doing in the lab was to start a company and engage in a drug discovery and drug development effort to bring that into the clinic. And so I think that for me, like one of the most important, intellectual revelations in my mind was that commercialization and translation are in many ways the same activity.
Yeah, I think that's a really interesting point. That's probably not the way that most folks think about it, right? That to actually do translational research, there is no other way because of how difficult it is to do drug discovery at academic institutions. And I can speak to this because in my lab, we're actually trying to get to the point where we can do some rudimentary drug screening. And MIT has tons of resources, and we are struggling to be able to really get like proper drug discovery microscopes and easy access to liquid handling on the scale that you need it. It's really stuff that doesn't exist so much in academic institutions. So you have to make a company to do translational research. Very cool. So you mentioned that all these things like race through your mind about do I file IP? Do I start a company? What was the actual first thing that you did to make this to make this a company, or to in your mind say I'm going to make this a company?
Yeah, so that's interesting. The very first step was I presented some of our work at an internal seminar series. We had what was called a immunology interest group. It was a place where we could go and sort of talk like internally on a rotating basis, present our science, right? And so I presented the story around the small molecule that we discovered that would selectively kill biomarker-defined tumors of an immunologic phenotype. And at the end of that meeting, the end of that seminar, one of our tech transfer people came up to me and said, I think we should write a patent around this. And that is the very first moment when it went from an academic exercise to an industrial exercise, to a biotech exercise. So the very first thing we did was we wrote a patent. And I had never been involved in preparing a patent before. I had no idea what was involved in it or what made a good patent or a bad patent. That one was a pretty good learning curve, right? There's a lot that goes into a patent, and I had to learn in a hurry that it's not quite the same thing as writing a paper or a grant, right? It's a very different kind of a thing. But once we had a patent filed and there was intellectual property, suddenly there was a potential to put a value on what we were doing, right? I mean, it's a theoretical value, but we were able to put a value on it. There was an asset that could be commercialized, and that asset is the intellectual property. And so it was then that I took the decision that the best way to take this forward was to start a company. So my next question was, how the heck do I start a company? I'm a scientist. I don't know anything about business. So I called a friend of mine, an old high school friend of mine, actually, who had charted a very different course in his career as a kind of a business guy, a management guy. And it was my first opportunity to pitch the idea to somebody who might be skeptical. And somehow I successfully pitched to him that he should join me as a co-founder in the company, and we could be, the scientist and the business guy, Tweedledee and Tweedledum, going out and building this company. He put a little capital into it. I put some sweat equity into it, and the next thing you know, we had incorporated a Delaware C Corp, and now it was a virtual company that was, for all intents and purposes, operating out of my garage, and that's where we got started.
Right. Wow. So I mean, it really came down to an old relationship with some business knowledge, and so. So you found this company, and what do you make a business model with your friend, and then do you start pitching, you start pitching people. Where did you go to, you knew you must need money to do something, to maybe get a lab space. What were your proximal goals to get this off the ground? And who did you pitch to try to get money to do those goals?
Yeah, so clearly we needed capital in order to be able to run the thing, right, and to be able to make any progress. But it wasn't just the dollars, right? We also had a really immature program and a really immature drug discovery asset, and we needed to raise value in the program itself. So there were two simultaneous goals. One of them was to go out and raise money, and the other one was to raise the value of the program by continuing to progress the science. And so a portion of the work that I was doing in the academic lab was around trying to deepen our understanding of the mechanism and to deepen the chemical biology, right? And so I was pursuing grant funding to enable what seemed like a bit more translational work than we had done at the time. At the same time, to build the company, we were out trying to raise funds that would enable us to bring the assets out of the academic founding institution and bring them into the company. And so very naively, we started reaching out to venture capital groups, to private equity groups, to family offices, to high net worth individuals, to angel investment syndicates, and pitching the company. And our original pitches were terrible, right? Bad. And we were told so. They told us this we don't believe it. We don't believe you. This isn't going to work. This is a loser. But over time, and doing that over and over again, we heard those. We heard that feedback. We refined the pitch. We refined the science, right? Because you hear in those pitches, what are the scientific questions that potential investors want answered? Right. Excuse me. And so we aligned the science with the biofinance a little bit, and it took a while. So we founded the company in 2012, and it took 3 years, a little over 3 years, for it to get initially financed. We got a little bit of friends and family financing early on. So in 2014, we pitched it to what are the labs there on Main Street?
Lab Central.
Lab Central, right? So we were. So with a sort of a partnership from J&J, they were just kind of getting Lab Central up and going. They were about half populated at the time, and they were running a pitch competition. And so we entered into that. And there were layers of process, and we got to know the people that were kind of making the decisions, right? We met with the first layer of people, then the second layer of people, the third layer of people, and they gave us a lot of advice on what works and what doesn't work, and we listened to that. And ultimately we're successful. So I think there were about 60 competitors, and we were one of 3 that won the sort of the golden ticket to space in Lab Central. And that was a big moment for us, because it was not only an opportunity for us to make the company not entirely virtual, but actually a physical company, but it also was hugely validating, right? That J&J and Lab Central believed enough in the story we were telling to put capital behind it. And that was a big domino that needed to fall. Once that domino fell, a lot of other doors opened for us. But for us to get that, it was a lot of stick-to-itiveness. It was a lot of persistence to get to that point, and a lot of failed pitches, and a lot of no's, and a lot of doors slammed in our face, but always taking that as kind of a learning opportunity to figure out what's going to be a little more effective the next time.
Right. You almost have to take the losses to eventually win. Cut your teeth. Uh,
Well, yeah, and I think you have to go into it with the expectation that your success ratio is going to be in the single digit percentages, right? I mean, you're going to pitch it a lot and you're going to hear a lot of rejection, and you got to have a thick skin. And like, we went into one pitch meeting and everybody that we were pitching to fell asleep, right? That's pretty tough to take. But we walked out of that and said, all right, something here didn't work. So uh, you just got to do it over and over again, and you don't know where things are going to stick, and you got to listen to them, right? I mean, they're smart and they're scientifically savvy, and they've seen this, and so you've got to listen to what their sensitivities and their scientific instincts are.
Can you just, because in the audience we have a lot of academic scientists and trainees, so I'm wondering if you can just briefly contrast what it's like to make a pitch deck versus a deck you might present at an academic conference.
Yeah. So in many ways they're very similar, and in some ways they're quite different. I think the effective scientific seminar, I think the most important thing in both cases is to tell a coherent story, right? It's not to do a data dump. It's not to download as much data as you possibly can. Sometimes less is more. It's really about being able to weave a narrative around your biology, your science, your technology, your concept. And in both cases, that's really critical, right? I've found that the most effective academic talks sometimes are the ones that are less data intensive and more about the narrative, right? Where you can get the main point, and there's just enough data there that you believe the main point. The same thing in a biotech pitch, right? It's really about telling the story of why is this a good company to back? Why is this a good technology to invest in? Why is this a good risk for you to take? I think one of the things that I've learned that's most important in both cases is to have a degree of honesty and realism about not only what's great, but also what are the limitations, right? So when you give an academic talk, you want to say, here's the conclusions we can draw, and here's the things we can't, and here's what's next. Same thing when you go to pitch a company to an investor, right? They hear hundreds of pitches that this company that's sitting before them is the next Google, the next Apple, the greatest thing since sliced bread, and they don't believe it. What they want to hear is what's good, but what's also bad, and what's your problem to solve the bad. And so I think, I don't know if it necessarily compares and contrasts the two sorts of presentations, but I think it's a common thread of what's really important to make a persuasive presentation, whether it's an academic or a pitch presentation, is to be fully scientifically grounded and realistic about what's doable, what's not doable, what are the limitations, what can conclude and what can not. And my experience with investors is that they highly respect the entrepreneur who can come in and speak honestly and openly about the limitations of their technology or the limitations of their idea, right? They view that as truth-seeking and as intellectually honest. And just in the same way that you would not want to write a paper and overinterpret the data, you need to be realistic about what the story you're telling is. And I think there's a tendency to try to oversell it to the VCs. And they all have PhDs in biology or chemistry, so they all know what you're talking about. So I think it's important to be rigorously honest about the data. Yeah. I think the other key, at least in building a company, pitching it for sure, but in building it also, is it's like the science is necessary but not sufficient. you've got to have a team. You've got to have a talented, quality team with the complementary skill sets to prosecute the idea. And this is something that I was told early on, right? Like a great team can succeed on a mediocre idea, but a mediocre team will kill a great idea, right? And I didn't really internalize that until we had started to build the company. And so I think the other part of pitching it, selling it, and building it is to put a great team around the idea. And we can come back to sort of how one does that, because it sort of seems like easier said than done. But I've come to firmly believe that you need to have terrific science and terrific people to do it.
Yeah. So I guess we can get into that a little bit when, in the context of you jumping full time into Cyteir. So I want to ask you a little bit about, where was the company when you jumped into being full-time CSO, and what influenced that decision? Was it that you felt you had to be leading that team to make it work? And then when you eventually left Cyteir, where was the company?
Yeah, sure. I mean, so when I joined the company in a full-time capacity, right, I was pretty aware that I was turning a page in my career, and that page was closing the chapter on my academic career and moving into a new phase, right? And that was a really difficult and soul-searching moment for me, because I had viewed myself as an academic always, and it took a long time for me to even contemplate a biotech career. And I think at the time I was thinking about it, I still didn't have a great sense of what that might mean for my own future, right? And so I told somebody at one point that it was a bit like jumping off of a cliff into a fog bank. But, in another way, it was a real evolution in my own self-identity. I was an academic, and to contemplate moving out of that and into industry was a difficult and soul-searching thing for me. But I was willing and able to do it because I also very much wanted to put the science that I was doing into the service of people. And I felt like doing it in this sort of basic research, grant-driven environment was, I could create that story in my head of how that helped people, but it wasn't very tangible. I couldn't directly draw the line of how the science that I was doing was leading to therapeutics, to cures, to diagnostics, to people who would benefit in my lifetime from what I was doing. And I increasingly felt like that was important to do, right? To be able to create new medicines for people who need them. And we just weren't doing that in the academic setting. And so, to be perfectly honest, the grants that I was writing and the papers that I was writing were more becoming increasingly oriented towards this translational, commercial sort of biotech space. And what I quickly found out, probably the hard way, is that there's a lot of enthusiasm for basic science, and there's a lot of enthusiasm for drug discovery, but within the confines of the federal funding agencies, there's not a lot of support for that thing that's in the middle. And I was finding my grant funding was dwindling, even though the company was taking off, right? And so I took that as a pretty clear message that where I needed to be was driving the drug discovery, right? And so the decision was a terrifying one, to leave behind this academic career that I always thought I was going to have. But it was also really energizing to move into this translational space where I could put the science into the service of people. And I had no idea what I was jumping into, none whatsoever, what it was going to take to make it happen. But it seemed like as terrifying as it was a no-brainer to make that move, because I wanted to create therapeutic applications out of the science that we were doing in the lab.
Right. So it really lined up with your passions in the moment. It just, yeah, it was obvious. So I want to let everybody in the chat know that I'll be keeping my eye out for questions for anyone who wants to ask them, but otherwise I'm going to keep asking Kevin questions. But if you have things that you want to ask, go ahead and drop them, and we'll incorporate them into the conversation. So when so you're at Cyteir and eventually you leave. So where did you bring the science to at Cyteir, and what made you want to find a new home?
Yeah, so the early days of Cyteir were really uncertain, right? We weren't sure where the next funding was coming from. We weren't sure if the idea was going to work. We brought in a great team of people that were really dedicated to trying to move this idea forward, and they did it. They were able to take this academic idea and bring it into the industry setting, be able to discover real molecules that really could be developable, do all the mechanism of action work around that, do all the preclinical work, do the IND enabling studies, get this molecule into the clinic. And now I'm proud to say that within just a few years of taking the company out of virtual mode and making it into a real company, we've gotten it into phase 1/2 clinical trial, and with encouraging results in the clinic, right? And so, as a basic scientist who was involved in the initial discovery of the idea that is now tangibly helping people in the clinic, like, that's heady stuff. That is very, very cool. And it's possible to do it over the lifespan of a project, right? I mean, we published the first paper in 2010 probably, and here 10 years later, which seems like a long time in a way, but 10 years later, we went from an academic publication to this thing is, patients are being dosed and they're responding, right?
Yeah. And
so that's extremely intoxicating. So as that project evolved, though, Elliot, you know me. I mean, my background is in the basic science and the discovery side of things, and I'm not a clinical researcher. I don't have an MD. I have not been involved in a clinical, science program, and I kind of felt like I really had the bug to try to what I really have the bug to do is to that kind of early stage discovery, that early stage biotech, that discovery to clinical kind of continuum. It's very cool to be involved in taking a crazy idea and turning it into something that actually benefits patients. And so as Cyteir began to evolve into a clinical stage company more, I had the hankering to kind of jump back into that riskier, more uncertain, kind of more churning early stage environment. And then it was very fortuitous, because around that time, as I was starting to think about these things, I met the founders of Civetta, Bill Sellers at the Broad and Eric Fisher at Dana-Farber. And within a half an hour of meeting those guys, I completely became enamored and seduced by the science, which is so cool. And so it was a really easy decision to make, to move over into that early stage environment again and sort of hand the reins off at Cyteir to somebody who had more experience on the clinical end of the development.
Yeah, it sounds like a perfect situation, right? Your expertise is less needed, and you run into these guys who no doubt have a really exciting exciting with technology to turn a company or to
Yeah,
make into a company.
and I would just, as a sort of piece of advice, something I've learned along the way, I think it's critically important to be smart and strategic and proactive about building your network. Right? Everybody says it, and everybody sort of knows that as a truism, but it's absolutely true that the people that you run into are the people who can be hugely influential to you in the future. Uh, I got introduced to these guys from a friend of a friend, a contact of a contact, and the next thing you know, there's this terrific opportunity for me, and it's a great fit for the company. Right? They needed somebody in my role, and I was looking for exactly the kind of thing that they had to offer, and it only ever happened. It only could have ever happened by networking. And so I think you can't overstate the importance of being able to build that network.
Yeah. So Michelle has a question about transitioning. Michelle, do you want to unmute yourself and ask Kevin your question?
Yeah, I mean, I think what's unusual about you, but also spectacular, is that you left relatively late academia, like you were a fully entrenched professor, whereas everyone on this call is much more junior to that, I think. So it's difficult every year I go further. I think that there's much more I need to learn and do within academia before transitioning. I'm sure most scientists also have that same feeling because we don't know what's beyond. So what sort of advice do you have about timing of transferring?
That's a great question. I think if you are passionate about the idea of being in the life sciences industry, then there's no time like the present to be able to make that transition, right? Because there's, the more experience you gain, obviously, kind of the how that's going to influence, it's going to influence the position that you move into. But to be honest with you, there's no experience like being in that environment to give you the qualifications that you need, right? You're going to come out of an MIT, Harvard, MD, PhD, with recognized as being very, very well trained, and then getting some industry experience under your belt is absolutely kind of the next sort of piece of the puzzle. One thing I do counsel people to do is, if you've got industry ambitions, think about doing a postdoc in industry. Right? These are there now in a way that they didn't used to be. And in my previous company, we offered postdocs, postdoctoral positions for people to kind of come right out of academia and gain experience. We have not yet done that in Civetta, but I'm definitely supportive and open to doing that, because I think, in a sort of a very practical kind of a way, when hiring is happening, one of the things they're looking at is what's the level of industry experience that you've got. Right? So the training is key, but your stock will be valued higher when you've got some industry experience under your belt. So if you're passionate about moving into an industry career, then there's no time like the present to do it.
Cool. So I want to ask you about just now that you've spent some time as a CSO, can you just sort of compare and contrast what it's like to sort of lead the scientific vision of an academic lab versus lead the scientific vision of a company? Like, how are those roles maybe more similar than someone this call might expect, and how are they quite different?
Yeah, it's interesting, right? I mean, I come from an academic background, and in a lot of ways, you'll never take that out of me, right? So I feel like my role in the company, both at Cyteir and at Civetta, is in many ways very analogous to your PI, right? It's to work with the group to establish scientific kind of vision and direction and kind of chart the course. Maybe not to create the detailed roadmap, but to sort of say, this is the direction we should go and that's the direction we shouldn't go. I think the other piece of it is to, kind of the business part of it, right? I have to pay a lot of attention to the business aspects of the science. How do we pursue science that will be commercially valuable? Will, and that doesn't mean, address the biggest market need, but it's like, what can we partner? Where can we create differentiation from the other players that are out there, right? I mean, there's sort of business concerns, and it's fundraising, right? And your PI does all those exact same things. It sort of set the overall direction, figure out strategically how do we differentiate ourselves from all the other people that are out here doing it, right? How do we publish the most unique and impactful papers, and how do I raise money? And those are the things that I do. Now, how those happen are a little different, right? Raising money is a bit more about pitching and partnering than it is about writing grants. But one thing, so sort of on that, just as a point on that, one thing, when I first made the jump from academia to industry, a number of my colleagues were like, don't you feel great that you don't have to write grants anymore? And the first thing is like, yeah, it's awesome that I don't have to write grants, but I still have to raise money, right? So I still have to go out and create an effective argument for the value of the science and be able to defend that argument in front of somebody who's evaluating it. So while I may not be writing grants, I'm still raising money, and you're raising money on the merits of the science. So I think there's a lot of similarities between a PI and a CSO.
Yeah, that's super interesting. So yeah, I'd love it if you could, just as we wrap up, if you could maybe touch on two things, like as you look backwards and forwards. So like, what are like some individual achievements? I know you talked about how seeing things in patients, but like maybe drugs in patients that you discovered. But maybe on like a little smaller scale, what are some things that you maybe personally achieved at Cyteir that you're proud of, and what, looking forward at Civetta, are you really excited about, either about the team or about the science? I know that I personally am really interested in the science at Civetta. I think it's really, drugging protein-protein interactions is obviously like a holy grail in drug discovery, and getting structural experts like Eric Fisher on it. It's just, I think it's a great concept. So I'm curious, sort of like looking backwards and forwards in your career, what are you proud of and what are you excited about?
Yeah, that's terrific. I mean, what am I proud of? A couple of things with respect to Cyteir. Number one, possibly first and foremost, is an idea that went from inception all the way into the clinic with, so far good clinical data, right? I mean, I feel unbelievably fortunate as a basic scientist to have been able to, with the team, conceive an idea that found its way into the clinic and could potentially help patients, right? I mean, that's a large, diffuse example, but I can't state strongly enough how unbelievably gratifying that is, to be involved in something that goes from nobody ever thought about this before to we're dosing patients and they're responding, right? So I'm super proud of the way the science has evolved, which brings me to the second part of what I'm really happy and proud of, is the team that we built to make that happen, right? That didn't happen because I sat in a room and had a great idea. It happened because there were a ton of people that believed in it and were creative and had ingenuity and were passionate about it, and all worked really hard through all the hurdles. And those are people both in the academic lab. I mean, Elliot, you know some of those folks. It's the people in the Cyteir laboratory who joined on a company at huge personal risk to themselves to join a startup company that had no definite future, but believed in the science and the idea, right? And I'm really proud of having been able to assemble that team, right, on a wing and a prayer. I feel like something I said to them was convincing. I'm not sure what, But you know, they joined, they stayed, and they made it work, right? And are still there, right? Past when I've left, and I think that's the third thing that I'm quite pleased about, is that, you know, there's a company here that has a long future, and I had the privilege of starting that company and being involved in its early days, and am sort of happy to move on and see it continue to thrive, right? To sort of be bigger than just my own ideas. That's really gratifying. That's very cool.
Yeah, for sure.
Looking forward, what am I excited about? Man, I'm excited to do it again. I just like, you know, the science at Civetta is so cool. It's different from stuff that I've worked on before, which is like intellectually invigorating and challenging. And like, I'm reading papers every day and like rereading them because I didn't get it the first time. And I'm working with,
so happy to hear that other people do that.
yeah, I like 3 or 4 times, and I'm still like, I feel like I need the Cliff Notes. And you know, and the people that I'm working with are amazing. The investors are great, and the people on the team are incredible. So what am I excited about? I mean, nothing short of having the opportunity to create, you know, novel medicines, to bring forward novel medicines for people who really need them, for whom there are not good options right now. And that's not a 2-year thing. That's not a 5-year thing. That's a 10-year, a 20-year, a 30-year thing. But I'm excited to be a part of that. Right? I would love to be able to look back, you know, 20 years from now and be able to say, Civetta and Cyteir both brought forward new medicines in novel areas, and people were helped. We saved lives. Like, that's super cool.
Yeah. It's the thing we've all written in our fellowship applications.
Right. And I wrote it every grant application I ever wrote, and now I get to live the process of doing it, and that's pretty cool.
Yeah, I mean, a ringing endorsement for all of us going into you know, entrepreneurial startup science, which I think, you know, everyone in this call is interested in. So thanks so much